Hey everybody, and welcome once again to our online lectures. In this opportunity, we will be discussing the third of the biochemical molecules. We're going to be talking about the protein. So let's jump right into our discussion. So proteins include a wide diversity of structures resulting in a wide range of function. Proteins account for more than 50% of the dry mass of most cells. So if you think about it, pretty much half of what you are, that's pretty much proteins. Some proteins can have like a functional or enzymatic kind of ability, and they can speed up chemical reactions. Other proteins, for example, have a function of defense, of storage, of transport, cellular communication, movement, structural support. Proteins are very, very active molecules that we have in the cell, and they work together to perform very, very distinct things in living things. So let's have an overview of the protein function. Like we said before, we have enzymatic proteins, and those pretty much, like we have been discussing in lab, they make chemical reactions go a lot faster. We have proteins that serve for defense, which pretty much protect living things against diseases by producing antibodies or producing antigens that can be used for detecting invading pathogens in a body. I have storage proteins like the storage of amino acids, like casein, the protein of milk, and many other proteins just have this functionality of saving things for later. Other proteins that are very, very, very important are proteins that serve for transport. And in the next chapter, we're going to be talking about cellular communication. This sort of proteins are involved in allowing things to move from one side of a membrane to another side of a membrane, or carrying certain materials and transporting them inside of the cell. Proteins can have hormonal functionalities. This type of hormonal functionalities are pretty much messengers that we have inside the body, and thanks to the action of those proteins, they can move a message or a signal from one organ to another organ. And this is how organs, we can say, talk to each other in a chemical way, to say it that way. They move messages around. I have receptor proteins, and they have a function or to response of cell to chemical stimuli, so they detect the messages. So hormonal proteins and receptor proteins, they work pretty much together one with the other, where the hormonal proteins serve as the message, and the receptor proteins are like the antenna of a cell phone that would receive this message so that that particular message could be interpreted. I have motor proteins, I have contractile proteins that allow contraction inside the cell, and a good example for this is muscle cells and the actin and myosin molecules, where they can make the cells shorter or longer, and if we look at this in a systematic way, it allows for movement. Other type of proteins that we have are structural proteins, and they have a main function of support. The example for this would be the collagen that we have in our bodies. They provide with flexibility. They provide that support that our skin needs to not be pushed away from one place into another. So as we can see, we have so many different functions for proteins, and it is to be expected, right? Because we are pretty much 50% protein. Then these proteins are going to be involved pretty much in everything from storage to communication to movement to structural support. Everywhere in the cell and in systems, looking at them in a systematic way, will be modulated or regulated by the functionality of proteins. So enzymes are one of the most important groups that we have been discussing for some time now. Enzymes are proteins that act as catalysts. And let's remember that the word catalyst, it's going to mean to speed up chemical reactions, to make chemical reactions move a lot faster, or even make chemical reactions possible. Uh, Otherwise, they wouldn't be probably possible. They need the action of an enzyme to for this to work. Enzymes can perform their functions repeatedly. They're functioning as workhorses that carry out the processes of life. Like we have said before, enzymes are like recyclable proteins. So once they get their job done, they can be reused again because enzymes are never consumed in a chemical reaction. So they are very handy things to have in living things because not only they make things move a lot faster, and chemical reactions like breathing and metabolism, they need to be faster. They are not used up in that reaction, so they can be repeatedly used over and over and over again. What are proteins made of? Proteins are constructed from the same set of 20 amino acids. So think about proteins or the available proteins as a work of art, where you have several different pictures in there and several different illustrations. How do I make that whole scene? How do I make that whole scene? Well, I need colors. So in order for me to make all of those different scenes in a work of art, the amino acids are going to be like the colors that I can mix together, that I can alter them, that I can mix and match to make pretty much an infinite amount of different proteins that I have. And I can do all of those proteins from just 20 amino acids. Polypeptides are unbranched polymers built from this amino acids. So if you imagine proteins as being a pearl necklace, the whole protein would be the necklace, and each individual pearl is going to be one of these amino acids. So I can use as much amino acids as I want, or as little amino acids as I want, and any combinations. I can take 20 amino acids of the same one, and that is going to give me a different protein. So just because of that, imagine the enormous amount of combinations that we can have from just these 20 amino acids. A protein, it's a biologically functional molecule that consists of one or more polypeptides. So this is like having larger proteins that are made not only of one pearl necklace, but of two, three, even four or more of these necklaces. So even more possibilities for different proteins. So we have a wide variety of this thing. A lot of these proteins. So amino acids are organic molecules with an amino and a carboxyl group. That is what makes this proteins pretty much proteins, and it differentiates what a protein is from a carbohydrate. Carbohydrates are made of carbon, hydrogen, and oxygen, and amino acids are made of carbon, hydrogen, oxygen, and and nitrogen, like we can see here in the amino group. So it's the amino group that's going to make the big difference in what a protein is. Amino acids differ in their properties due to differing side chains called the R groups. So if we look at amino acids, if we look at the 20 amino acids that we have, they're all going to follow pretty much this same structure, and the thing that it's going to change is going to be this extra group that we call the R group. So I'm going to have pretty much the same organic molecule with this R group that will differ between those molecules, and that is going to make me the 20 different amino acids that I can have. In terms of their structure, how do I make them? Amino acids are linked by what we call covalent bonds, and they have a special name because they are for proteins, and we want to differentiate these bonds from any other type of bond. They're pretty much a covalent bond. We call it peptide bonds. And a polypeptide, it's a polymer, and let's remember the word polymer. Polymer, it's a molecule that it's made of a sequence of repeated similar molecules, again like a pearl necklace. So a polypeptide, it's a polymer of this amino acids. Polypeptides range in length from a few of these amino acids to almost 1,000 of those different monomers. And remember the combinations. It could be in any combination, so I could have almost an infinite amount of proteins. Each polypeptide has a unique linear sequence of amino acids with a carboxyl end, which we call a C terminus, and an amino acid that we call an N terminus. And here we have a representation of what happens in here, and this is pretty much the dehydration reaction or the hydrolysis reaction that puts together these bonds. Let's remember that hydrolysis means breaking in the presence of water. So if I have a big amino acid and I add water, I should say a polypeptide, and I add water and the appropriate enzyme, I can break down or separate that chain of polypeptides by hydrolysis. And by dehydration, what means is that I will take the hydroxyl group and the hydrogen of another amino acid, and I can join them together. That releases a molecule of water, and it creates a brand new covalent bond that in the case of amino acids we are going to call a peptide bond. So we are applying what we learned from the chapter in chemistry about our covalent bonding, but now we are adapting that to biochemical molecules to understand how do I go from a single monomer into a polymer that consists of many, many, many different monomers. And this is the example for my proteins. Okay? The specific activities of proteins result from their intricate three-dimensional architecture. Remember what we have said since the beginning of this class, that proteins or anything biological, its function would be determined by their shape. Structure determines function, and in proteins that is exactly what the case is. The specific sequence of amino acids are going to determine the shape of the protein, and therefore it's going to determine the functionality of that enzyme. So the shape, it's very, very important. A functional protein consists of one or more polypeptides precisely twisted, folded, and coiled into a unique shape. So now that we understand the shape of a protein, we can see or we can apply things that we have learned from our lab, that temperature can change or alter the bonds that make up this protein and therefore change its shape. And changing the shape will change the functionality. Same thing with the pH. Chemical things that can alter chemical bonds have the ability to alter the shape or structure of a protein, and that will alter its functionality, which could be giving it a different kind of function or inhibiting that protein altogether. Okay, there is this is a little animation of how proteins will pretty much work, and you can download this presentation to your computer if you would like to see this animation, but for the sake of time, I am going to skip ahead and continue talking about my proteins. So the sequence of amino acids determines a protein's three-dimensional structure. That's something that we have to keep in mind to understand how these proteins work a little bit better. Shape is everything in the functionality of a protein. A protein structure will definitely determine how does it work. The function of a protein usually depends on its ability to recognize and bind to some other molecules. So even though now we are thinking about chemistry, chemistry in a way has to do with physical things. So this is the way that we're going to look at proteins now. Chemical things are physical things. So a protein has to physically attach to a substrate so that it can do the function that it is intended for the protein to do. And that functionality, again, and I know that I'm repeating myself on this, but that functionality is going to depend on the shape or the structure of a protein. A better way of thinking about this or analyzing this with more common things of our daily use, it's like a screw and a screwdriver. If I don't have the proper size or the proper or the indicated screwdriver for a screw, the work is going to be a lot slower, or maybe I won't be able to put the screw in or take the screw out because of their shape. So imagine if you have the proper screwdriver for the proper screw, and then you hammer the tip of that screwdriver and you alter the shape. Now it won't be able to do what it was intended to do. So even though it's the exact same screwdriver with all of its components, its shape is going to be a lot different, so it is not going to work. That's the way that we should think about proteins. That's the way that we should visualize the proteins in order for them to work. Okay? The primary structure. Let's talk about the levels of protein structure, because now we're talking pretty much as when the protein is being created, and how does the protein starts bending, it starts twisting until it finds the proper three-dimensional structure that it's necessary for protein function. First, we start with what is called a primary structure. This will go primary, secondary, tertiary, and finally quaternary. The primary structure of a protein, it's its unique sequence of amino acids. This would be amino acid 1, followed by amino acid 2, followed by amino acid 3, and so on. You get the gist. This would be like a linear polypeptide that will only include the proper order of the amino acids. The secondary structure, it's found in most proteins, consists of coils and folds in the polypeptide chain, and this was going to be determined, of course, by the functional groups and the chemistry, the repulsions and the attractions between those groups. This is going to create folding, and this is going to create coiling of the proteins. And this is what we're going to call a secondary structure. When I have a coil or a folding in the protein and nothing else. A tertiary structure is determined now by the interaction among various side chains, or the R groups. And in a tertiary structure, we're going to see that we have both of those twisting and the folding and linear structures, and they start coiling and bending onto itself, creating kind of like a three-dimensional weird shaped ball. Imagine you are playing with a piece of paper. So when the paper is brand new, it's talking about a white piece of paper, a blank piece of paper, I should say. When it is completely new, that will be the primary structure, and then a secondary structure would be if you roll the paper or if you start bending the paper, like making folds on that paper, one or the other. But imagine if you start bending this paper and then rolling this paper and crushing it between your hands, you're forming like a ball that has a weird ball shape with folds and twists, and this is what a protein is going to look like. So what is a quaternary structure then? A quaternary structure results when a protein has multiple polypeptide chains. So I have, in other words, I have like two smaller proteins that are together to perform a function. It has subunits of protein. So I can have two proteins, three proteins, four proteins, all together to create a larger complex of protein that will have one function. And this is what's the quaternary structure of a protein it's going to be. So let's think for a second how much combinations I can have from this protein. Think about the complexity of life. And this is one of the reasons why we have so much complexity in life. I can have any combination of any length of 20 different things, and I can bend this thing to my liking. And not only that, but I can take different proteins that I can make in the exact same way, and I can put them together to make something completely different from the original two proteins that I have. That is how much combinations of this thing I'm going to have. So let's have just a quick look, a visual look at the structures or the levels of my protein. This is a primary level structure of a protein. As you can see, it's like a line that has a bunch of amino acids, and it is just the sequence of those amino acids all being in there together. This is one or the other is what the secondary structure of my protein is going to be. Here we have the coiling of the protein, and this is called an alpha helix. Helix because it's kind of like a round, twisty thing that happens with the proteins, and we can see the hydrogen bondings that are forcing this coiling to actually occur. The folding of the protein, it's called the beta strands or the beta folds, twist the proteins up and down, and they're also together by their hydrogen bonding. Hydrogen bonding with other part of the polypeptide is going to create this bending motion. So if I have one or the other, this is what I call a secondary structure. So when in a protein there is several secondary structures together, then I have this three-dimensional structure, which is what I call a tertiary proteins that contains both alpha helixes and beta plates all in the same protein. So there we have that three-dimensional kind of like structure. And now finally I can look at what a quaternary protein would be. So it would be, in other words, and to put it very simply, two or more tertiary structured proteins that are working together towards a common goal. Like, for example, hemoglobin, it's this type of protein that contains several subunits, and let me show you that in here for a second. I have several subunits. I have four um, subunits, both with tertiary, quaternary structures, and these molecules together, they are going to perform the function of what hemoglobin actually is. And the interesting thing about hemoglobin, just to put it out there, because I know that you might be thinking about it, I've heard about this word before. How does it work? What hemoglobin? What it does, it transports oxygen when you breathe it from your lungs through your blood to the rest of your cells, and oxygen attaches to the heme group of hemoglobins, and all those hemoglobins would be inside of all of those would be inside of the red blood cells. Okay, so moving here my slides a little bit quickly. Let's go to this other one here. The primary structure of a protein, it's just the sequence of amino acids. This is just putting a little bit more detail into each one of these individual things. It's just the sequence. The primary structure, it's like the order of the letters in a word. Primary structure is determined by inherited genetic information. And later we're going to talk about this transcription and translation process. How do I know the order in which a polypeptide should be formed? Well, that is going to be coded in the DNA of an organism. Depending on the order of the letters of the DNA, that is going to be the order of the amino acids of any protein that I have in the human body. That is why DNA is there. It's the recipe for making all of these structural things. The coils and folds of a secondary structure result from hydrogen bonding, like we saw in the picture before, and the coils called alpha helixes, and the folds are called beta plated sheets. In the tertiary structure, that is when I have a three-dimensional look at my protein. All of this is created by hydrophobic interactions, van der Waals interactions, everything that we discussed as weak forces, and those very, very strong covalent bonds that I have there joining my protein together are disulfide bridges, and they reinforce the protein structure. And like we said before, this quaternary structure would be a combination of tertiary structured proteins that are working together for a common functionality. Let's have a look at what happens, for example, when things are not correctly, when things are not right with the functionality of my proteins. A slight change in a primary structure can affect a protein's structure and ability to function just by changing one amino acid. Going even deeper, changing one letter on the DNA of an organism can lead to a different protein that might not work at all. For example, sickle cell anemia, which is a disorder that it's a blood disorder that it's inherited, results from a single amino acid substitution in the protein of hemoglobin. And that abnormal hemoglobin molecule causes the red blood cells to aggregate, to like clump together into chains, and then deform into a sickle shape. And here we have that. Sickle cell anemia, it's a variation of the sixth amino acid in that sequence, and what that makes, it creates abnormal hemoglobin, and they kind of like clump together, like we can see here, kind of like in a tube, and that causes the overall shape of the red blood cells to change completely. And people that suffer sickle cell anemia have different problems, including weakness and diminished ability to transport oxygen to their organs and many other things, just by changing one amino acid. That is the result of changing one letter in the DNA sequence of a person. So what determines the structure? Denaturation and renaturation of a protein, or changing the shape and bringing that shape back. In addition to the primary structure, physical and chemical conditions can alter or affect the structure of a protein. For example, changing the pH, changing the salt concentration of a solution, temperature, or any other environmental factors can cause a protein to unravel. It causes the protein to lose the secondary structures in the protein, and that is going to definitely lead to a loss of functionality. When a protein is no longer functional because of a change in their structures, we call that denaturation. And a denatured protein, it's biologically inactive, so it doesn't really work. Depending on the change, depending on what caused the change on my proteins, sometimes that denaturation can be reversed, leading to a renaturation of that protein. Very interesting. It all depends on how much that it's being altered. So how do we get to understand? How do we know? How do we discover many of these things? By scientific exploration, seeing what a protein does, if I inhibit that enzyme, what chemical reaction is no longer being generated, and so on and so forth. Scientists would use X-ray crystallography to determine a protein structure, which is pretty much using X-rays shot through a crystal to create some sort of an image that we can later on use for approximating the shape of this protein. Another method is nuclear magnetic resonance, or NMR. This is type of spectroscopy. Bioinformatics, it's another approach to the prediction of a protein structure from amino acid sequences, which involve reading this, pretty much reading all of the amino acids, their order, analyzing their DNA, because we're later on going to go into a lot more detail about how do I get the order of a protein, but it involves the order of the letters of DNA, and it involves translating that information into another language that organelles, which are the ribosomes, can use to put the amino acids in the correct order. So proteins are very, very interesting proteins, very interesting molecules, I should say, very highly specific. They are very picky with how do they work. They are completely dependent on the information of DNA, and without a doubt are some of the most important biochemical molecules that we can have. Not only about half of our body is made of proteins, but proteins are involved in every single cellular process in one way or another. It storing something, a message, transporting a message, contracting, moving, carrying things around, moving materials from one place to another. Proteins are definitely at the center of every source of biological action. On the next video, we are going to discuss the final of the four biochemical molecules. We are discussing the nucleic acids. So thank you for your attention, and I will see you on the next video.